everyone, my name is Regina Spranger. I'm a PhD candidate at UC Santa Cruz in the Ecology and Evolutionary Biology Department. Today, I'm excited to share my work with you on the future of North American amphibians. This is a story of ecophysiology, acclimation potential, and conservation. When we think about the future of amphibians under climate change, there's a lot more intricacies than just if organisms can survive increasing temperatures. Think about this frog hopping through the forest. To understand how it will be affected by climate change, we must think of other environmental conditions besides temperature, like change in hydric conditions, increased fire frequency, and canopy density. But there are also things like the thermal tolerance range of the animal, how an animal may adapt in its lifetime, if there are generational effects, how genetic diversity impacts adaptations, and so much more. In my research, I aim to understand the true extinction risk of amphibians by discerning these details. To do this, we first study ecophysiology. We investigate how environmental conditions like canopy density or humidity affect microclimates and those cascading effects on amphibian thermal physiology. Secondly, we study how much an organism can acclimate within its lifetime. Acclimation is a form of plasticity that reflects how the environment impacts an individual's traits, either morphologically or physiologically. Acclimation is more than just an animal surviving a new environmental condition, but actually physically adapting to it and returning to baseline traits. Thirdly, we must also consider acclimation potential that comes from other sources, like from different life stages or between generational acclimation. We measure between generation acclimation potential by seeing how maternal effects and egg incubation temperature affects offspring thermal physiology. With this information, we can calculate the cumulative acclimation potential over generations. I can unite all these projects to understand the large-scale physiological patterns of amphibians facing climate change, clarify the extinction risk for these animals, and show how we can use this information in conservation projects. For example, we can use our understanding of ecophysiology to determine if a canopy coverage is too dense for frog thermoregulation and have land managers trim the canopy. Or in my current project, we could use our information on thermal physiology and acclimation potential to test for outbreeding depression between populations. This could help U.S. Fish and Wildlife prioritize conservation efforts in our local amphibians. With my work, we can continue adjusting our conservation efforts and better predict the future of amphibians. Thank you.